We are Team Beta from Puiching Middle School, Macau. Today we would like to share our innovative research topic, supramolecular aggregation and fusion of lysosomes. So what's our project mainly about? So here's a cell, and this is a lysosome. It performs cellular defense by digesting bacteria. We binded MOR-PEG-ADA to form MOR-PEG-ADA. To synthesize MOR-PEG-ADA, PEG-ADA was firstly synthesized through the dehydration condensation reaction, and MOR was bonded with PEG-ADA through a condensation reaction. We conducted HNMR study on our molecule to evaluate its chemical shift and analyze whether it has been correctly synthesized. On the next step, we binded CB7 and HA together to form HACB7. We prepared the samples for antibacterial assay and confocal study. Prior to the antibacterial assay, we prepared different concentrations of bacteria samples. Bacteria was incubated in an agar plate and stored at 4 degrees Celsius for daily or weekly use. A colony of bacteria is transferred to a test tube with an inoculate needle. The extracted bacteria was incubated in 2 ml of lysogeny broth medium in a test tube and placed onto the shaker at 37 degrees Celsius to maintain the suspension of bacteria. After 12 hours, bacteria was extracted to conduct further experiments. To prepare the samples, cells were firstly placed into an agar plate with DMEM in an incubator for 12 hours. MOR-PEG-ADA and EGFP E. coli was added into the agar plate for 4 hours. Subsequently, we removed the medium and added different concentrations of HACB7 for 6 hours. The medium was then removed and replaced with lysogeny broth medium. With a cell scraper, we broke the cell membrane and released the bacteria from within. The bacteria was transferred into a test tube and brought to the shaker for 12 hours. For the samples of confocal study, the cells was placed in dishes. Once the samples was ready for observation, PBS was firstly added to each dish to rinse all the waste products from the cells so as not to interfere with the observation. MOR-PEG-ADA and EGFP E. coli was added for 4 hours. Subsequently, we removed the medium and added in different concentrations of HACB7. After the preparation of samples, we were able to start operating the system. To obtain a more accurate ratio-wise image, oil was immersed on the objective lens to change the refractive index. Subsequently, the specimen was placed on top of the objective lens. After all, we set the adjustment knob to allocate the cell. Lastly, we click the shutter to save the image. This is the result of the confocal study. To perform colony counting, 4 different groups were placed into the LB agar plates. 100 microliter of different groups of diluted bacterial solutions were added to the plates respectively. The solutions were then spread equally using glass beads. After 24 hours of incubation, we counted the numbers of colonies and checked the antibacterial effectiveness of each group. This is our result of the bacterial colony counting experiment. With the addition of MOR-PEG-ADA into a cell, lysosomes could be targeted. By adding HACB7 and MOR-PEG-ADA bonded together through host gas effect and pulled lysosomes proximally. In this way, our project was able to induce the aggregation of lysosomes.